is that this is the connecting stalk. The outer part and the inner part of the outer segment, right? This part of the plasmalemma is continuously undergoing enfoldings, which is leading to the formation of these membranous discs or these membranous lamellae, which give the transverse striation appearance in both the cone and in the rod. In the case of the cone, there is still continuation with the basal plasmalemma, but in the case of the rod, the membranous lamellae become independent of the surrounding plasmalemma. And these discs are continuously moving distally, and as they reach the distalmost portion, they get removed from the outer segment. And what happens to them later? They are then engulfed by the retinal pigment epithelial cells, which are also considered as scavenger cells in this. So that is one of the functions of the RPE. This was, as I've said, this is the connecting stalk. Then we have the inner segment, which is responsible for the metabolic function. For example, it produces the phospholipids and the proteins needed for the formation of the plasmalemma, for the formation of the membranous lamellae. Then we have the outer rod fiber. Now in the case of the cone, the outer rod fiber is almost non-existent, and there is almost direct continuation with the pericardia, right? Whereas in the case of the rods, the outer right fiber is a structure in its own right. You can quite see it prominently, and then there's the pericardia. The pericardia is basically the part where the nucleus is located. And these two actually are part of the third layer. And over here you can see the outer nuclear layer, and this was the outer rod fiber. Now we will have the inner rod fiber, which is actually very long in the case of the cone, but rather short in the case of the rod. And then we have a terminal dilation known as the cone pedicle, kind of like this, and a rod spherule. So these are all of the portions, right? But the thing is that after this, it's not quite so simple. In addition to this is the terminal portion of it, and this is the connecting portion of it, right? This is, my apologies, this was the inner rod fiber, and this is the inner rod fiber. This is the axon, and this is the axon, and this is the terminal dilations, the spherule and the pedicle. And these actually lie in the fourth layer, which is the outer plexiform layer. Until the outer plexiform layer, the blood is derived from diffusion via the choriocapillaries. So I'm hoping this kind of makes sense. Then we will move towards the inner nuclear layer. In the inner nuclear layer is the pericardia of the bipolar cells, and they have these dendrites coming up, right? So this part is also, actually all of this is the outer plexiform layer, because in the outer plexiform layer you have the synapses, and you have the axons of the photoreceptors, and you have the dendrites of the bipolar cells, and you also have, actually this is, as I've said, where this is present, it is the inner nuclear layer, right? In the outer part of it, we have these cells known as horizontal cells, and these horizontal cells, they interact, they have these synapses with the rods and the cones, so they connect those two. And these are the horizontal cells, and they are found in the outer part of the inner nuclear layer. And there are types of them. There are the diffuse horizontal cells, and then there are the midget horizontal cells, right? So we have types of those. My apologies, my classification was wrong. I kind of messed it up. I was right that the connection is between these, and generally this is the dendrite, and it is one cone, and the other end tends to synapse with multiple cones and rods, the axon. Then in the outer part of the inner nuclear layer, we will have the endocrine cells, and we'll consider their function at a later date. The thing you need to know is that their pericardia is in the outer part of the inner part of the inner nuclear layer, and that they don't have any axon, which is a key factor, and they have many branching dendrites which pass into the inner plexiform layer. That is the only thing you need to be able to understand at this point. So the third function of RPE is that it is going to be storing the vitamin A, which is needed for the formation of rhodopsin and iodopsin, and when photosensitivity needs to be increased or decreased, there is back and forth exchange between these and these. So that is the third function which we need to be considering. Functionally, frequency wise, we have many more rods than we have cones, and the rods are much more light sensitive. That is also another thing to understand. Then we talk about the bipolar cells. We have types, we have the diffuse, and we have the midget, right? And we also then talked about so my apologies. So these are the bipolar cells, my apologies. So we have the diffuse and we have the monosynaptic. Diffuse, and it will be interacting with multiple photoreceptors, and this would be synapsing, this axon would be synapsing with multiple ganglia cells. But in the case of the monosynaptic, just a cone, not a rod, just a cone and just one ganglia. That would make it a midget bipolar cell. So after this we have the fourth layer, fifth layer, sixth layer. The seventh layer is the ganglia cell layer, where there is a cell bodies of the multipolar ganglia cells. Right, and then their axons, basically they pass into the last layer, the layer of optic nerve. Right, and these will eventually form the optic nerve when it will exit at the optic disc. So these are some important things.